As she mentioned, my name is Pryce, and I'll be discussing workflow orchestration in bioinformatics with an open source tool called Flyte. So just a little bit of background about myself and my journey through the orchestration landscape. I got a degree in molecular biology from UW, just up the road. In my senior year, I took a course by Mary Claire King, who described the BRCA gene a few years ago now, and she really inspired me to pursue genomics. From there, I've always had a knack for computers and CS, so I self-taught that side of things and wanted to bring them together into bioinformatics quite stubbornly. I harassed people on LinkedIn until someone gave me an opportunity, and I've been in the industry ever since. Finally landed on an open source project that I was building myself back in the day that needed some orchestration. I started out with quite an exhaustive evaluation of different approaches and landed on SnakeMake. It's very approachable, and I love SnakeMake, but I ended up needing more container orchestration features, so I evaluated Docker Compose and decided to just bite the bullet and learn Kubernetes. So from there, instead of trying to shove SnakeMake into Kubernetes, I was again evaluating workflow orchestrators. I was probably going to settle on Airflow with the Kubernetes executor, but landed on Flyte, which I kind of stumbled upon, and it has a really fabulous UI console that sold me. So I dove headfirst into the Flyte. It has a vibrant community and ecosystem. I was very active in there. And a few things that Flyte emphasizes that I'll get into more later are type safety, language independence, although the main SDK is in Python. Almost everything, all Flyte workflows and tasks that live today are written in Python. It lets you declare infrastructure right alongside your task and workflow code. And finally, it abstracts the data persistence layer so you don't have to worry about your data flow. Finally, from Flyte, I went to Union, the company where I now work that maintains the Flyte open source project as well as the community. So just a brief primer on Flyte concepts. We have tasks, which are very self-explanatory, the basic unit of work, but a few things I need to call out here. The inputs and outputs, as I mentioned, are strongly typed. So you have to declare them alongside in your Python functions, as you can see here on the left. And those are really required for the workflow to be composed at compile time. These are true Python functions, and that is just a task decorator. There's more to that decorator, as we'll see in some later slides, but that's the basic setup. Things are also, or all tasks and workflows, pretty much every concept in Flyte is versioned when you register it. So that's a nice automatic sort of reproducibility bonus. And of course, as a Kubernetes native executor or orchestration layer, everything is containerized and runs in its own pod. Workflows, of course, are what you compose tasks into. Again, the inputs and outputs of workflows themselves are strongly typed. They're also versioned, as I mentioned. And something to call out is that Dataflow really is a first-class citizen in Flyte. Some other orchestrators kind of leave the data flow as a bit of an afterthought, and you have to deal with it yourself. Here, the object store is tightly integrated into the execution layer. So it really is how defining those inputs and outputs between the tasks is how the workflow is inferred, which gives you a number of benefits. The workflow code here on the left is not a true Python function. It's actually a domain-specific language that gets interpreted by the Flyte engine at registration or compile time. But you can see those two tasks that I had in the previous slide. Greet is just being called, and then you're passing the output of greet into say, just with that greeting argument there, and that's pretty much how you infer a workflow. So I need to discuss some of the challenges we have in bioinformatics before getting into it to motivate the problem. Of course, I'm sure many of you went to the keynote. You know about the massive volume of data that all scientific disciplines are generating at the moment. Bioinformatics, or biology in general, is not spared from that. Since the sequencing of the human genome, there's just been a fire hose of data, new wet lab processes have been automated, there really is more data than we can hope to analyze currently. Of course, the subject of this track is languages playing nice together. Bioinformatics tools are all across the board. You have your C, your C++ for the low-level, heavy lifting. Java is super popular. The picture here on the right is actually the human genome printed out on a bunch of books in like 4-point font. It's enormous, but that's courtesy of the Broad Institute. They put out what is arguably the gold standard of genome analysis tools, GATK. That's all Java. Python, of course, is wonderful, as we all know, but it's not necessarily super performant. I'm sure we've beat that drum a few times here. Everyone loves R for visualization. Rust, of course, is entering the fray, bit of a darling. And then the new ML models like Deep Variant, AlphaFold are all very exciting at the moment. But all these languages have different dependencies, different package management paradigms, compiled versus interpreted. It's a whole mess. Additionally, we often rely on a traditional file system. In bioinformatics, you expect certain files to exist in a directory with certain extensions and a certain header structure, perhaps. That doesn't necessarily play nice with modern database solutions, for example, or if you're operating in the cloud, you're usually using an object store, especially if you want to save some money. So making those things play nice together is super important. So in short, these are examples of the same challenges across the scientific community. They really boil down to reliability, reproducibility, and ease of use. So next I'll go over just a brief primer of a very common part of a workflow, at least in bioinformatics, called alignment, and then discuss some of the key ways that Flyte solves some of these challenges. So alignment, for those that are not bioinformaticians among us here, is super, super common. Basically, we have really long strands of DNA in all of our cells. In order to do analysis in any meaningful time frame, we parallelize that dramatically by chopping it up into smaller pieces. The sequencer will essentially produce massive text files of these smaller chunks that we now call reads. And then we want to realign those reads to a known reference genome so that we can essentially add back in that location information so that we can do meaningful downstream analysis from there. So a very common sort of first step in any analysis pipeline. So the first way that Flyte seeks to solve these problems is a concept that we have called image spec. So this is a dependency management solution. You can think of it as an opinionated reimplementation of a Docker file. At the end of the day, it will produce an OCI compliant image depending on the builder that you use. But really it ships with support for, at the moment, PyPy, Conda, and apt packages. But of course, there's also arbitrary run commands for pretty much anything else that you want. The point being that it's trading a little bit of flexibility for some convenience. I'm sure many of us have struggled with setting up, maybe pulling in MicroMamba, for example, setting up the right environment, installing your packages, making sure that environment is available when you run your image, for example. Image spec handles that all for you. You just tell it what you want in the final product, and it'll do it. It also has swappable builders, so you can use Docker, EMVD. You can even have remote builders so that you basically ship off this image spec to wherever your Flyte cluster is running, and it'll build it for you, so you don't have to wait for your local machine to build and push that image to a repo. An example where this is really nice is you could take any image off the shelf and just say you want a FlyteKit dependency in your image spec, and it'll build it for you and make sure that everything, all the Python path is set up correctly. The next way that we're tackling the playing nice problem is with these data classes or data types. So again, I mentioned that bioinformatics tools often rely on a traditional file system. There are some challenges there. And we have an example of those reads on the bottom left here in red, the characteristic TG, CNA. So this is a snippet from a FASTQ file. Like I said, it's just a really long text file. We're not that fancy in the bioinformatics world, oftentimes. But that's kind of the basis that we all standardize around at the moment. And if you have a bunch of these FASTQ files, for example, in an object store, being able to capture them in a data class and make sense of them with additional metadata and standardize the naming convention makes using them a lot simpler. A few more things to call out here. Since I mentioned Flyte is strongly typed, so you can pass this reads data class as an input or output of your different tasks. We're also capturing inside of the data class the FASTQ files as Flyte files. This is a Flyte-specific type that is aware of the object store, so it'll handle serialization and deserialization into and out of it for you. What was I going to say? Finally, yes, safety. A lot of times, if you have a big shared file system, you can have side effects that are unintended for one experiment, maybe clobbering the outputs of another experiment. Flyte, since it is just merged to an object store, all the file systems are completely ephemeral, so you have to explicitly say, I want to pull in these inputs from the object store, I want to serialize these outputs to a unique prefix in that same object store. Any unintended side effects just go away when the pod gets deleted. So the final and most important way that Flyte helps all these different languages and tools play nice together is that we have a common orchestration layer inherent in the tasks. We have a few different ways of defining these tasks. Python tasks are the most common, they're the most feature-rich. We also have shell tasks, which allow you to just define a script that you want to run inside that pod. We finally have container tasks as well, which are similar, but they don't require any FlyteKit dependencies whatsoever, so you really can just bring your own container and run it in a task. These are two tasks, the shell task and Python task, that are doing different parts of that alignment workflow. Bowtie 2 is a very common memory efficient fast aligner, and we wrapped the first part, or the first task, in a shell task to produce a Flyte directory, and this first task is generating an index from that known reference, which is a required first step for any alignment to perform efficiently. Different aligners will have different indices, but the point is that we're making this Flyte directory that has that index in it. Then you can see that we're passing the index into the align paired reads Python task there for Bowtie. It's that same Flyte directory. And we're also passing in that reads object that I described earlier and producing an alignment data class, which is similar to the reads data class, but slightly different, obviously, downstream. A few more things to call out in the Python task. We have in FlyteKit a subproc execute, which is basically a Flyte-aware subprocess run. And the reason we're putting it in this task like this, instead of just using a shell task, for example, is within a Python task, you have a lot more flexibility. You can unpack data classes before processing, rebuild them afterwards, make whatever paths you want. So you just have a few more options than in the shell task. And I also want to draw your attention to one of Flyte's key strengths here, in that at task decorator, I said there was more than meets the eye. That's where you can define what container image you want to use. So that's the image spec that I specified a couple of slides ago. You just pass it in, main image, and when you go to register the workflow, it'll build that image and push it to the whatever registry you have configured and use it in that task. So you can use different images for different tasks in your workflow. And finally, the declarative infrastructure comes into play here, where we're defining the resources we want for this specific task. So here we have CPU and memory. These map one-to-one to their Kubernetes equivalents. So you also have things like GPU. You can just say, I want 4 GPUs. And the Flyte engine will handle that for you. You can even go down to the accelerator. Say you want a very specific architecture for your GPU for this task. You can specify that right there in the task decorator as well. So finally, we come to put this all together. I've omitted I haven't gone over some of the tasks in this workflow, but this is where we're defining the workflow itself. So we're starting off with a directory containing a bunch of FASTQ files, and then we're producing a Flyte file. That's the whole workflow. At the beginning, we start with fastqc, which is a common quality control tool written in Java, and that'll just produce a report of different QC metrics for us. We also have a convenience function or task called prepare raw samples, which is just to generate those reads data classes instead of from a Flyte directory by just parsing through it. So you can have an arbitrary number of samples that you want to run over. Next, we're going to do some filtering with a tool called fastp, which I've wrapped in a Python task. So it's pyfastp. And here we're actually passing it to one of our parallelism constructs called a map task, which essentially fans out and runs that pyfastp over however many samples were present in that input directory. Next, we're going to explicitly define some task dependencies, so you don't have to rely on the inputs and outputs flowing into each other to create a workflow. You can explicitly say, I need this to depend on some upstream here, and the reason we're doing that is making sure that filtered samples is dependent on fastqc in case fastqc fails, for example. We don't want to continue wasting compute resources on bad input data. Moving along, we're actually getting to the meat of the issue here by generating that index using that shell task I had on the previous slide. So we're passing in a known reference genome. Reflock is just a location in the object store, so it'll pull it in from the object store and generate that index for you. And then you can see bowtie2 aligned samples, pulling it all together with the index and the filtered samples. And the astute among you may have noticed that bowtie2 aligned samples is not bowtie2 aligned paired reads. So aligned paired reads is the singular version, whereas bowtie2 aligned samples is using another parallelism construct we have in Flyte called a dynamic workflow. Very similar to a map task. It's a bit more flexible, but it's a bit less performant. So all of that to highlight, we have a few different features for working through something fairly simple. And finally, we're calling the render multi-QC task that I didn't go over, but this is using multi-QC to generate a final report with all the run statistics of everything above it. So we're passing in the fastqc output, the filtered samples, which contain reports themselves, and the alignments, which also contain reports. And multi-QC generates a really lovely HTML report, which we're then saving as the output of the whole workflow as a Flyte file for saving it longer term. So I know that was a fair amount. I went through a lot of different Flyte features, workflow constructs, task types. It's fairly simple to tease this apart from just the code, but the Flyte console that brought me to really appreciate Flyte in the beginning has some really wonderful visualizations. This is a couple of them. Of course, no workflow orchestrator would be complete without a DAG somewhere. We have what I all just went over, fastqc, prepare raw samples, feeding into the map task there, which is for some reason really tiny, but it's only doing it over one set of samples. At the same time, the bowtie2 index is being created. All of that is feeding into the align samples dynamic workflow. That's why it has those start and end nodes, because it is another workflow construct, but you can compose workflows sort of a bit more arbitrarily in Flyte, so you can just throw a dynamic inside of a top-level workflow. And finally, everything is flowing into render multi-QC to create that report. The bottom view is a timeline view, which is really nice for visualizing individual task executions in the overall context of a whole workflow run. Some other things to call out here are fastqc and the bowtie2 index were actually read from cache. So Flyte has caching as sort of a first-class citizen. You just declare it right alongside that task decorator. Again, you can add a cache key. There's a few different approaches to it, but it's very granular, and it's really powerful for things like an index, which for a given aligner isn't going to change very often. And then fastqc, I just read it from cache because I was experimenting and didn't want to have to go through that step every single time I was debugging something. Everything's green because it succeeded. These are color-coded, though. If they failed, it would be red or empty for downstream tasks that didn't complete or didn't run because something failed upstream. Yeah, that's a couple of views from the console. So the whole point of all this is to highlight how many different languages we just blasted through in that fairly simple workflow again. The pre-processing tools were written in Java and C, C++. The alignment tool Bowtie 2 is implemented in Perl and C++. The reporting tool MultiQC is purely written in Python. And finally, I included Ferris the Crab because I like him. There was no Rust in that workflow, but he's really cute. Yeah, so that's pretty much it. That's how Flyte can help these different tools play nice together. It's really wrapping them in a number of different ways into this common orchestration layer, and then enforcing the different types they pass between each other in a very rigid way. Thank you.
Thank you, Pryce. Questions?
Hey there. Nice to see you again.
Hi, Prof.
This is a question I asked kind of just offline yesterday, but I figured I'd ask it again kind of for the benefit of everyone else in the room. Could you maybe provide some understanding of like how Flyte differentiates itself a little bit from a lot of other players in the space like Kubeflow or MLflow or Airflow?
Mm-hmm. Yeah, so there's a lot of people or a lot of orchestrators built on Kubernetes. There are a number of orchestrators built with Python as the main SDK. I think the main ways really are going back to sort of the first slide that I had there. They are strong typing everywhere, so you get those compile time guarantees ahead before wasting any compute. The data flow abstraction, being able to just forget about the fact that you're using an object store underneath and just focus on your workflow code is really powerful. You don't have to do any Boto3 or S3FS calls, for example. And then finally, it's that infrastructure declaration, being able to just say what you want and then sort of leave it up to the infrastructure people to make sure they have the node pools required to run that, but being able to keep all of that sort of in one place alongside with your image spec definitions, for example, and have that all just in one versioned area, basically.
Other questions?
So to expand a little bit on that, is Flyte kind of implemented in the same level of the stack as your Kubernetes implementation, or does Flyte guide your Kubernetes implementation? Could you describe that a little bit?
Yeah. So Flyte runs on any vanilla Kubernetes cluster. So GKE or what's Amazon's one? Oh my God, I touch it every day. But yes, it's just built on top of any vanilla Kubernetes infrastructure, and then it implements FSspec is used for all the files and directories. So anything that FSspec supports, like the different object stores, it'll talk to those. It's not necessarily trivial to set up a Flyte cluster. I mean, it's not trivial to set up a Kubernetes cluster and maintain a Kubernetes cluster, but it is definitely doable. There's guides for it. That's all open source as well. So if you wanna go through it, it's approachable.
One more. Thanks for acknowledging the Flyte deployment pains. Somebody that's actually gotten Flyte deployed on-prem, it's great to hear somebody else say it. So one question I had to do with, it's a bit of a two-parter for map tasks. If one subcomponent fails within that map task before like a reduce, is that whole thing considered a fail? And then the follow-up to that is, does Flyte have the capacity to just rerun failed tasks, not whole workflows?
Yes. To answer your first question, map tasks will say you have 1,000. If 100 fail, that's fine. You can actually specify a failure threshold in that map task for when you want the whole map to fail or pass. And your second question, I'm sorry, what was it?
Yeah, so does Flyte have the capacity to just rerun failed steps, not whole workflows?
Right. Absolutely. So there is a concept of retries with failures. User errors, so like if you just code it wrong, it'll fail immediately. System errors, you can preset a number of retries that you want. Say you or something, you can retry that automatically. But then the user errors, we kind of bias towards failing fast instead of wasting compute. However, with the caching that I mentioned, having that granular cache mechanism, and as you saw with my FastQC task, that workflow failed a few times before it was all green. You can just cache everything up to there. And in fact, we have a few customers that do this with some really massive fan-outs. They put caching everywhere, and anytime something fails, they just hit rerun egregiously until it goes through. That's one way of doing it.
So we have time for one more question.
So thanks for the great talk. So I noticed you have image spec, which solves the problem of making sure the various libraries your workflows are going to depend on. For example, if it's a Python task, arrive at the pod. Mm-hmm. But frequently, looking at your code that you had here, which I understand isn't the complete code, it didn't look like you had the import statements inside the functions. How do you manage, getting the environment the same that you're coding locally versus what gets shipped?
So that is a common pattern, putting those import statements within the tasks. And that would be a pattern that I would use locally. I rely so much on Bioconda that I've completely given up on trying to use it locally on my ARM OS X machine because it just doesn't target it. So I actually use image spec to generate those images sort of as one-offs or at least as smaller self-contained units, and then I run all my tests inside of a container locally. So you get a little closer to the production deployment. I mean, there's a million different ways you can do this, but yeah, 100% you could import there. Running inside the container, I'm allowed to have those imports at the top of the module.
Let's thank Pryce again.
Thank you.
We'll continue.